Good afternoon, everyone, and thank you for joining us for today's webinar. Today's webinar is entitled Investigative DNA Testing Implementation. This is the final webinar in our SNP DNA webinar series. We have three fantastic presenters today. Our first presenter will be Dr. Catherine Gettings. Dr. Gettings began working in the field of human identity testing in 1998 and obtained her Master's of Science in Criminal Justice, specializing in forensic science from Virginia Commonwealth University in 2001. She trained and worked in the Forensic Biology and DNA Casework section of the Virginia Department of Forensic Science and later became the technical leader of forensic operations at Bodie Technology. She has testified in numerous criminal cases. She has taught forensic science courses at the undergraduate and graduate levels, and she has served as the Forensic Laboratory Auditor Assessor with NFSTC and ASCLD Lab International. She earned her PhD in Biological Sciences from George Washington University in 2013, and then began her postdoctoral work with the Applied Genetics Group at NIST. Our second presenter today will be Ms. Moretta Chase. Ms. Chase has worked at the Center for Forensic Sciences for 11 years, the past 6 of which as a senior technologist in the DNA unit. Prior to joining the CFS, she worked at the Analytical Genetics Technology Center at Princess Margaret Hospital, doing SNP genotyping using microarrays and mass spectrometry. She received her bachelor's and master's from the University of Toronto, where her master's focused on the role of adhesion proteins during fruit fly development. Our final presenter today will be a little special. We do have Dr. Runa Daniel for a recording that she had done previously. Dr. Daniel is a research scientist in the Office of the Chief Forensic Scientist at Victoria Police Forensic Sciences Department in Victoria, Australia. Her primary research focus is the development of forensic DNA intelligence capabilities and emerging DNA technologies. She has been engaged in massively parallel sequencing applications in forensic DNA analysis since 2012 and co-chairs the Australia-New Zealand Massively Parallel Sequencing Scientific Working Group. She holds an honorary appointment in the Adjunct Associate Professor at the National Center of Forensic Studies, Faculty of Education, Science, Technology, and Mathematics at the University of Canberra. All right, our disclaimer real quick. The opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. Now, before we do get started, I would like to hand things over to Dr. Daniele Podini for our opening remarks.
Thank you, Josh. Hello to all of you out there. It's my pleasure to welcome you to the last of the series of the webinars of this series titled All Things SNPs. We are finishing strong today with a great panel of speakers that will focus on using SNPs to predict the biogeographic ancestry of individuals. We will talk about the technical and analytical aspects of the process, but also will address implementation of such assays and discuss how the results of these tests could be communicated to investigators and how investigators can and will be using this information. This latter aspect has not often been discussed, so I'm very happy that we are able to include it in this series. So that said, let's get started. Our first speaker is Dr. Catherine Gettings.
All right, thank you, Daniele and Josh. Thanks to all of you who are listening in today. So as Daniele said, we'll be talking a little bit more about the application of the ancestry SNPs and some implementation issues today. I too have a disclaimer that I have to share with you. Any opinions that I state are solely my own, and I will be identifying some commercial equipment, instruments, software, and that does not imply an endorsement. So hopefully at this point in the webinar series, this acronym is familiar to you, that SNPs are single nucleotide polymorphisms. The human genome, as we know, is about 3 billion base pairs, and each genome has 4 to 5 million variant sites, and over 99% of those variants are SNPs, or short indels. That varies by population, how many SNPs you have, and there's a really beautiful paper referenced here from 2015 from the 1000 Genomes Project Consortium that looks at SNPs and the variations in SNPs in populations around the world, and I'll show one other image from that paper shortly. So most of the SNPs that we're talking about are biallelic. That means they have 2 different states. So within an individual, you could have 2 different types, meaning you'd be heterozygous, but you could also be homozygous for one allele or homozygous for the other allele. So 3 possible genotypes. And this paper is about 10 years old now from an ISFG SNP panel discussion, and it laid out 4 different categories of SNPs. So there are individual identification SNPs that were covered in a previous webinar, the ancestry informative SNPs that we'll talk about today, the phenotype informative SNPs that were also covered in a previous webinar, and we'll touch on them a little bit today, and then the lineage informative SNPs. So those are Y chromosome SNPs or mitochondrial DNA SNPs. That could be a piece of the overall ancestry, but it only gives you one line or one lineage of an individual's overall ancestry. So I want to go over the II SNPs just as a counterpoint to the ancestry SNPs. So the individual identification SNPs are ones in which a balancing has occurred in all the populations. Ideally, the allele frequencies will be similar within and between populations. So that means they should have a low FST, if you're familiar with those population genetics terms. And they should also have a high heterozygosity. And there are a couple papers cited here at the bottom from Dr. Ken Kidd's lab. These are the II SNP panels, or panel that is included in the commercial software, one of which, one of the assays, commercially available assays that I'll be talking about later. And then the ancestry informative SNPs are kind of the opposite. They're ones in which a population-specific fixation has occurred, and they should have a low heterozygosity, so they should be all the same allele within a population. And that's going to be different for one population. So in the example here, we've got in the continent of Asia has one genotype, and all the other continents would have a different genotype. So if this was a SNP, it could give you continental level ancestry. Some examples of that are malaria resistance in sub-Saharan Africa. There's a SNP RS2814778 that is associated with resistance to malaria. And so there's one genotype that is really commonly found in sub-Saharan Africa that can be used in that type of ancestry determination. And then also lighter skin pigmentation in Europe. So a lot of the SNPs that you heard about in the phenotype webinar are ones that we can use to predict a European ancestry because they might be associated with blue eyes or lighter skin pigmentation in Europe. When we talk about heterozygosity, I want to show just quickly how we calculate that. If this is a population of individuals with each circle being a person and the letters within that circle being their genotype at a particular SNP, we can calculate the allele frequency just by adding up all the A's and the G's and dividing them. And so this is 60% A and 40% G. And from that, we can calculate the genotype frequency with P squared or Q squared or 2PQ. And the 2PQ value at the bottom there is the heterozygosity. So in this case, the heterozygosity is 48%, and that's about the best. The best heterozygosity you could get would be 50%. So this would be a really good individual identifying SNP if this distribution was present in all the populations around the world. And now on the other hand, this would be a good distribution for an ancestry informative SNP. So we have population Z over here. And we have all of the individuals, almost all of the individuals are of an A genotype. We have one person here who's an AG heterozygote, okay? And then over here we have all the other populations, A, B, C, etc., where everyone is GG homozygous. So this SNP would be good at differentiating population Z from all the other populations in the world. And that's how most of these SNPs work. They're good at distinguishing one population from all of the others. This is the other image that I mentioned earlier from that 1000 Genomes paper. We have such a wealth of information now in the public domain about SNPs and their distribution around the world. So what we can see from this image is, we look at this pie down here, this is our key. If we look at the dark gray part of the pie, it says shared across all continents. So these are SNPs that are found in populations and continents all around the world. And you can look at the distribution of those SNPs in populations on the map here. And so those are the pieces of the pie where you would want to look for individual identifying SNPs, because those are present in all populations around the world. If you're looking for continental level ancestry prediction, you would look at the lighter colored piece of the pie over here that's private to a continent. And if you wanted to predict individuals from a specific population, you would look for the darker colored piece of the pie. So you can see populations around the world have different size pie pieces, and that relates to kind of human migration patterns and population bottlenecks that have occurred. But just having all this information at our fingertips now is really valuable for forensic purposes. All right, so moving into the actual assays, I think of the Ancestry SNP assays as having three components, and so I'm going to break those down. The first is a panel of target SNPs. So that's something that would be published in the literature. The researcher has developed this panel that's good at a particular differentiation. So a panel might be good at continental level ancestry differentiation, or it might be doing something within a continent to break apart different populations. But it's important for a forensic lab to understand what question they're trying to answer, what populations are most common where that lab is present. That would be the populations of interest. And so for the commercially available ancestry prediction, ancestry SNP panels, there are two panels that we'll be talking about. And one I call Selden 128, and that's for the anchor author of this paper, Michael Selden. And this was published in 2008, so about 10 years ago. It's called Ancestry Informative Marker Sets for Determining Continental Origin and Admixture Proportions in Common Populations in America. And the self-described purpose within that paper is to provide a method for determining and quantifying differences in continental populations. The application, as described in the paper, is to evaluate participants in association studies due to ethnic disparities in disease risk. Okay, so it was not designed for forensic purposes. They looked at 825 individuals from European, African, Native Admixed American, East Asian, and South Asian populations. And they were looking at only the SNPs that were present in the SNP array that was in wide use at that time that had 300,000 SNPs. Nowadays, we have SNP arrays that have millions of SNPs in them. So this was a relatively small number of SNPs that they were looking at and a relatively small number of individuals. And like I said, the purpose was not forensic in mind. And then the other panel that is included in some of these commercially available assays I'll call KID 55. It comes from Dr. Ken Kidd's lab, and it was published more recently in 2014 in this paper here. And the purpose, as described in that paper, was to provide a method for identifying the geographic ethnic origin of an unknown sample. And it was really designed as a forensic investigative tool. They also explored many databases. The difference between the availability of information from 2008 to 2014 is enormous. So they looked at 650,000 SNPs from 1,000 individuals from 51 populations. They also looked at a number of SNPs and individuals from samples that are housed in their own laboratory, and other large data sets, including the Selden 128, were evaluated. These are images from those two papers. So the top one came from Selden 128, and the bottom is from KID 55. These are the structure plots. So structure seeks to take a set of genetic data and break it down into a user-defined number of groups, or populations in this case. So these are showing a pretty big difference in what these assays are doing. Now, some of the difference is how the structure analysis was set up. So in the top analysis, the number of populations was set to 5, and in the bottom, I believe it was set to 8. So that's part of the difference here, but another part of the difference is the population level information that's available for all of these KID samples allows you to break them down and look at how well those SNPs are actually differentiating very finite and discrete populations. So I think what I would draw from this is that the assays were designed to do two different things. And I like to show this. This is a totally different paper, but this is also from 2008 using a 300,000 SNP array. The authors were showing how they could resolve populations across Europe. And I think if we think about a map of Europe and we look at the different markers or for the different the legend here, the data here largely recapitulates a map of Europe. And so I think the point I like to make with this is that we can do a finer level of differentiation, but we have to know what we're looking for, and we have to design the assay and the interpretation model with that in mind. All right, and I briefly want to talk about the assays that are now available in the instrumentation. And for this, we're largely looking at the same types of questions that you would look at in any validation. So when you're looking at a lot of different loci at the same time, you want to look at the interlocus balance to see if you're routinely expecting to get results at all of the different markers that you've included. We want to look at the heterozygote balance. So are there any genotype-specific biases present? And just concordance in general. So concordance across replicates and concordance to other methods. This first image here is based on data that we evaluated using the forensic assay, and so that's run on an Illumina MySeq FGX. And that assay contains 172 SNPs that are meant to help with ancestry and phenotype, but also identity information. So those might be useful in a case of degraded DNA. If it's too degraded to obtain an STR profile, the identity SNPs might still be able to perform. Those are all being assayed at the same time as the STRs. So this 172 SNP panel contains the KID55 for ancestry. It also contains the H-IRISplex SNPs that Dr. Susan Walsh spoke about a couple months ago. The data that you're seeing here on the left side y-axis is the average SNP coverage, which is ranging from 23x to about 3500x. And on the right side y-axis is the average heterozygote balance, which is ranging from 0.42 to 0.94. And perfect heterozygote balance would be 1. So we can see from this type of view that there are some SNPs that are performing better than others, and that's something that we would want to think about in terms of the validation, what our expectations are, and how that impacts the statistical analysis. This is the Precision ID Ancestry SNP panel. This contains 165 SNPs for ancestry. So that's containing the KID55 and the Selden 128, and there is some overlap between the two. And this can be run on the PGM instrument or the S5 instrument. So again, on the left side axis, we have the average SNP coverage ranging from 264x to about 2000x, x being coverage or copies of a consensus sequence. And the right side y-axis is again the average heterozygote balance. It's about the same range, 0.43 to 0.98. So some of the SNPs are performing better than others, and that's what we see in these very large multiplexes, is that it's hard to get a perfect balance that we're used to seeing in a smaller STR panel or even in a snapshot SNP assay, where we can take the time to balance each SNP or each STR. If there are only 20 or 30 of them, that's a much easier task than trying to balance something in an amplification of hundreds of SNPs or hundreds of markers. All right, and then the last part of this that we want to spend the most time on today is the data analysis and the interpretation model. So, assuming that we've chosen a good panel of SNPs and we have an instrument that can correctly genotype them, then this is a really important piece of this puzzle that the practitioner needs to understand, because this is where you provide the link to the investigator. I think Runa will talk a lot more about this later. The predictions, just like choosing the panel of SNPs, it depends on where you live and what populations are of interest. The predictions are also going to depend on having the right model training data. So the Precision ID Ancestry Analysis is the panel that consists of KID55 and Selden 128. And there's a manufacturer's analysis plugin that uses Alfred population data, and that's also coming from the lab of Dr. Ken Kidd. The method evaluates the expected occurrence of the evidence profile in each Alfred population. The number of populations will vary by set. We're talking about KID55 or Selden 128. But just to give you a sense of it, there are 125 populations that have been typed in KID55. So it is a large number of populations, and you'll see some of the output that you get from that. You get a number for each population. This gives us the potential for a very specific ancestry prediction But that brings with it other challenges that we'll talk about in a moment. This is what some of the output looks like. And for anybody who's familiar with this, this is an older version of the analysis software. So I'll point out one difference later on, but it's largely the same. But if you notice a difference, it's because this is an older version. It was designed for the PGM. So if you're using this panel on an S5, it might look a little different. So you can see that we have results tabs for the KID 55 and also for the Selden 128. And we have the actual SNPs and the genotypes. And then we have a results tab that gives us, for each of the populations that are present in Alfred for that panel, it gives us a likelihood. So that's the probability of finding that profile in that population. And so if it is the population from which that profile originated, then that should have the highest likelihood of finding that profile. And you also get a heat map. So this just gives you a quick at a glance where the likelihoods are largely focused, where the lowest likelihood numbers there are focused on a map view. And then you get the same thing if you click over to the Selden list of 128, you also get a view like that. So when I'm showing data later on, I'll show two maps. I'll show you the heat map from the KID 55 and also from the Selden 128. You can see some of those side by side. And then the forensic, the ancestry and the phenotype SNP analysis. That panel is the KID 55 for ancestry prediction, and then 22 phenotype SNPs for the phenotype prediction. The software is using 1,000 genome data to give us continental level ancestry prediction. So the interface looks like this. Excuse me. And the phenotype information at the top there is based on the H-IRISplex system. So you're seeing a hair color result and also an eye color result. And then the ancestry prediction is based on the 1,000 genomes data. And the plot for the ancestry is a principal component analysis, where you see the first component here is largely differentiating. The colored dots that you're seeing there are the 1,000 genome samples. So the first component is largely differentiating the African population from everyone else. And then the second component is largely differentiating the East Asian population from everyone else. You can see a red dot there, and that is the sample that we actually ran right here. And that is grouping well within the European population. And that's correct for this individual. You can also see that the phenotype prediction is blonde hair and blue-eyed, which is also correct. But you can also get a sense from this that you're not getting a very fine level of resolution on the ancestry part of it, and there's not much else you can do with this interface. But you can, I'll show you in a moment, that you can take this data out of it and put it into a couple of freeware tools that might be useful. So I want to take you through, with the remaining time, a number of samples. These are samples that we collected at GW when I was working on my PhD here with Dr. Podini, and show you how those individuals group with the genotyping done on the PGM and using their interpretation software, and also with the forensic software. So this is an African American individual, and we can see with the PGM, the KID 55, it's grouping well within kind of Eastern Africa, and with the Selden 128, it's grouping more toward Western Africa, but still, we would consider that correct. And then with the forensic, we're grouping right within African American. So this looks just like we would expect it to. An East Asian individual, similarly, is grouping where we expect it to for the KID 55, the Selden 128, and also for the forensic. Now we get into some more interesting things when we have admixed individuals, and that's really common in the United States. So I think it's incumbent on the laboratory to test individuals during their validation that are challenging like this, that might give different results. It's not that helpful if you validate just with all European individuals, for instance, and you might miss a lot of the challenges that you'll have in interpretation later on. So this individual is half European and half African American. We can see that the KID 55 is grouping them kind of in a South Asia over here, and the Selden 128 is grouping them in Africa. And the KID 55 with the forensic interpretation model is grouping them kind of halfway in between the European and the African populations. So none of this is right or wrong necessarily. You can kind of think about reasons why population genetics and human migration and all of that. You can think about reasons why this might happen and look this way. It's just important to understand what to expect when you're working with unknown samples. This person is half European and half East Asian. So again, they're grouping, on the PGM over here, they're grouping kind of halfway between Europe and East Asia geographically, and that's what their genetics also look like. In the forensic, they're grouping in admixed American, which would be what we would think of as Hispanic. But it is, on the plot, it is halfway between the European and the East Asian populations. That just happens to fall within the admixed American. We think about principal components as having multi-dimensions, not just two dimensions. We think about this plot having a third dimension. It's possible that that third dimension would resolve this ambiguity here. It would resolve the fact that this individual is not Hispanic, but in fact that they're half European and half East Asian. So some of this might be a limitation of the resolution of just the visualization of the data. Now, this is a Hispanic individual, so let me toggle back to that half European, half East Asian. Keep an eye on that red dot right there. And now let's look at a Hispanic individual. You can see that they're grouping at almost the same spot. So, like I said, I think that would be challenging to differentiate the two with this method. And also with the method with the heat maps, it's also very similarly grouping kind of geographically halfway between East Asia and Europe. And so, running enough samples in your laboratory and understanding these challenges is key. Now, with the PGM or with the S5 data, however you're generating that data with a precision ID, you get all of these different likelihoods, like I mentioned earlier. And I would think in a laboratory, you would want to come up with some guidelines on what these numbers actually mean and how to report them. And so this is just an example of how you might categorize that data and deal with it. So one way to handle it would be to take anything that's within a factor of 1,000 and to say that all of those things are equally likely, because based on your validation data, based on running a lot of samples, anything that you're seeing in that grouping is equally likely. And that's a way to kind of deal with this resolution of data. And so if you did that, you would be making a statement in a report that would be something like the profile is equally likely to be found in a Central Asian, Native American, or European individual, right, based on the geographic regions that are shown here. So that might be marginally helpful, but I think the more helpful statement part of that would be that you might be able to say it's over 1 billion times more likely if the person is one of those three things, European, Central Asian, or Native American, versus than if that person is East Asian or African. So you've essentially ruled out two possibilities, even if you can't precisely say what population they're from. And one of the things actually that's in the newer version of this software is an admixture component. And so there is a little bit more help within the software itself now for distinguishing this type of admixture. And the last thing that I want to share with you are two freeware tools that you could use if you've generated this type of data, if you want to explore it on your own and do some things that the manufacturer of software might not let you do. So the first one is called Snipper, and the website where you can find that software is right here at the top. This is coming out of the University of Santiago de Compostela. That's the home of Dr. Chris Phillips, who spoke a while back about the II SNPs. And they have some really nice tools here where you can take the genotype data from either the PGM or the S5 or the MiSeq FGX. You could take that data out and run it through their analysis methods using training sets that are already available on their website for these different panels. So the Snipper method, it allows you to choose a few different classifiers. You could use a Bayesian, multinomial logistic regression, or genetic distance classifiers. So you can really try different things with your data. There's some good visualization tools there too that would allow you to work with the data in a much more hands-on way. And then the other open source tool that you could use is FrogKB. And so for the PGM or S5 data, it's already using the Alfred population. So FrogKB should give you something that looks very similar to what you're getting in the Thermo Fisher application. But you could do things like if you wanted to simulate dropout or simulate profiles, you could do that here and enter whatever you want to and see what the result would be. And then you could also take that MySeq FGX data over here and get the training set for the KID 55 and work with that data and get that same type of output that you can get from the PGM or the S5. You could get that from your forensic data as well. So I would encourage labs to really understand the data. It's not, the challenge is not generating correct genotypes. The challenge is interpreting the data and sharing it with an investigator in a responsible way. So hopefully that's given you some food for thought on the different aspects of these assays. That we start with a panel that's typically been developed by a researcher. It's important that that panel targets the right populations for where you live, where your lab is located. And then there's an assay and an instrument. And for those, we're considering the same types of questions that you would normally consider in a validation. And then there's also the data analysis and interpretation model that can have a very large effect on the final result. The predictions are very dependent on the model training data, and it's up to the practitioners to responsibly translate that information for the investigators. And with that, I'd like to thank our funding sources, or the FBI's Biometric Center of Intelligence, also NIST Forensic Sciences, our Special Programs Office at NIST, and the National Institute of Justice. And I'd like to acknowledge my group leader, Dr. Peter Vallone, for all of his help, and also the people who did this genotyping for the actual data that I showed today in our laboratory are Kevin Kiesler and Becky Steffen. With that, thank you all for listening, and I'll turn it back over to Josh.
All right, thank you so much, Dr. Gettings. We are going to go ahead and hop right into Moretta's presentation. Once this populates, Ms. Chase, if you want to go ahead and take it away from here.
Sure. Hey, hello everyone. I want to thank the Forensic Technology Center of Excellence for asking me to give a talk about our validation of the Precision ID Ancestry Panel. So this is one of the panels that Catherine spoke about, so I'm going to give some more details on it. Here's an outline of my talk. I'm going to first give you an idea of why we targeted Ancestry as a service to provide to our clients. And then I'm going to go into the instrumentation, chemistry, and our mode of interpretation or analysis method. And with that, I'm going to talk about some of our Ancestry testing results, and then the reporting guidelines that we currently have under consideration that we developed from these results. And then finally, I'm going to touch on some forensic type samples that we ran through the system and what the results look like. Okay, so first, just a little background about our lab. So I'm with the Center for Forensic Sciences, and this is a provincial government lab, forensics lab, that serves the approximately 13.6 million people in the province of Ontario. So we have two locations, one in Toronto and the other is in Sault Ste. Marie. And we're accredited by ANAB and the FBI QAS system. So currently in our biology section, the main service we offer is SCR typing, and we do this with the ID Plus panel. But as those of you are aware, currently in cases where the target SCR profile has been generated, but it hasn't been associated with anyone, either by direct comparison to a specific person of interest or by comparison to a convicted offender from the Canada's National DNA Databank, we've basically reached a dead end until we can find that relevant comparison. So to help bridge that gap, the CFS is targeting ancestry testing. This service is also in direct response to the interests expressed from our clients. And it's also critical to note that the province of Ontario is populated with people from a wide range of ancestral backgrounds, like particularly in our urban centers. And our hope is that by providing our clients with this ancestry information, we can be providing an investigative aid that will help them in cases where there's no suspects, and it will help them prioritize tips. So the instrumentation we're using for this is Thermo Fisher's Ion S5 massively parallel sequencer. This is our first time using the instrumentation. This is our first service we're offering with it. We also have the Ion Chef, a robotics instrument, which basically it automates our MPS workflow all the way from library preparation all the way to chip loading with minimal hands-on time. And as I mentioned already, the chemistry we're using is the Applied Biosystems Precision ID Ancestry Panel. And as Catherine mentioned, there is another method or model of interpretation of the data, and that is the admixture prediction. So Catherine talked about more the population likelihood method of analyzing the data, and my talk is going to be strictly around the admixture prediction method. Okay. So I'm going to give you a little background about how the admixture prediction method works or model works. So there's 146 ancestry informative SNPs that are used for this model, and they were sequenced in a number of populations across the world, and these black pins indicate their approximate geographic locations. So Thermo Fisher took this data and grouped the populations based on the similarities of their allelic frequencies for these SNPs into 7 root populations. So we have the Americas, Africa, Europe, Southwest Asia, South Asia, East Asia, and Oceania. So what the software will do is it will take the genotype data of your unknown sample and try to find the best combination of those 7 root populations to describe the sample. So that's how it's an admixture prediction. So it's trying to find multiple ancestries to explain that person if necessary. I also just want to point out there's some black pins left in the map, and those populations, we refer to them as intermediate populations. They didn't, when Thermo Fisher was doing their clustering, these populations didn't cluster with any of the 7 root populations. And they basically found that if they were to include them in those clusters, that people of admixed or multiple ancestries would cluster with that grouping. So obviously the software does all of its calculations, and then it will spit out a result. And this is what the results look like for the admixture prediction. So at the top you have a map which gives you a quick visualization of the ancestral breakdown of the person. So in this case, this person declared themselves to be 100% Filipino. Below the map, all 7 root populations are listed with a breakdown of the contribution of each of those root populations. So in this case, their prediction is coming back as being 90% East Asian and 10% South Asian. So 2 things to note along with this is that the maximum number of ancestries, root populations that will be reported is 4, and that the percentage go up in increments of 5%. Okay, so now I'm going to go through some of our results. So before I go on, I probably will alternate between talking about admixture prediction and ancestry testing interchangeably. This part of the talk is going to, we're looking at, we obtained samples from people who declared themselves to be 100% from one particular root population, and we were testing to see how accurate their prediction was compared to their declaration. We asked people to declare their ancestry back as far as they could. Most cases we tried to get it back to their great-grandparents, but it's always something to note that in this testing, we are depending on their declaration, and not always do people know their entire ancestry. So that's always something to keep in mind. Okay. Okay, so as I said, these are people who declared themselves to be from 100% from one root population. So I'm going to talk about the Americas populations in the following later in the presentation. We didn't test any people from Oceania, so that leaves the remaining 5 root populations that I'll talk about. So if we go all the way, oh wait, I got my little arrow here. Here, if we look at this group is the people that declared themselves to be 100% East Asian. So we tested 10 samples, and their results were between 80 and 100% East Asian. So this particular group had the greatest agreement between what they declared and their ancestry results that the software predicted. If we move over to South Asia, you can see that range is broader, so their results came back between 50 and 90% South Asian. So the people that had a lower South Asian prediction, the remainder of their prediction was basically European or Southwest Asian or a mix of the two. And then if we move further over to Southwest Asia, you can see the range was even broader still, so it was between 25 and 90% Southwest Asian that their prediction indicated, and the remainder of their prediction was European if they had a lower percentage of Southwest Asian in the prediction. So the final 2 populations I'm going to talk about in a little more detail. So we'll start with the African individuals we tested. So you can see their prediction, the percentage of their prediction that was African was between 0 and 100%. So if we break that down into geographic locations, we can make more sense of the data. So the two people that we tested from Egypt had African prediction between 0 and 10%. The people that we tested that were sub-Saharan Africa, their prediction was between 85 and 100%. And the 5 unrelated Ethiopian samples we tested were between 30 and 40% African in the results. So if we look at the populations that were used to define the region, we can better understand these results. So the orange pins over here are the populations that were used to define the African root population, and you can see they're all sub-Saharan populations. So it makes sense that the sub-Saharan samples we tested had greatest agreement between what they declared and what was predicted. You can also see that we have these 2 intermediate populations which fall in Ethiopia and Somalia. So once again, it makes sense that our Ethiopian samples came up as an admixture. They appeared to be an admix of African and, in this case, of Southwest Asian. The Northern African samples, the 2 Egyptian samples we tested, their prediction was predominantly Southwest Asian. And that, too, we can understand better if we look at the early migration pattern of people coming out of Africa, the first wave coming out, and then we can see that Northern Africa was populated by people coming from Southwest Asia at a later date. Okay. So we can do the same with our 100% European samples and divide them into Northern Europe and Southern Europe, and you can see that Northern Europe has the greatest concordance between what's declared and what was predicted. And once again, if we look at the data underlying the root population, we can understand why. So you can see all the blue pins are the populations that were used to define Europe. And then the pins that are in black are Southern European populations, and they are those intermediate populations that weren't used to define the region. So the Southern European samples that we tested, we saw that they had a European prediction between 55 and 90% European. And those with the lower European prediction, the remainder of their prediction was Southwest Asia. Also to note, 2 of the populations in that intermediate group were 2 Jewish populations. So we also tested 8 people of Ashkenazi Jewish ancestry, and their European prediction was between 45 and 100%. And those with the lower European prediction also had the remainder of their prediction was Southwest Asian, similar to the Southern European samples. Okay, so in summary, just because that was a lot of information, I'll just give you a little summary here. So the samples with declared East Asian, Sub-Saharan Africa, or Northern European ancestry had the greatest agreement with their ancestry results. We did see people who appeared to be admixed, and so that was the groups from Southern Europe, the Horn of Africa, as well as South Asia could appear to be an admix of those, the root populations they're closest to, along with Southwest Asia. South Asia could also appear to be an admix with Europe. And then finally, Southwest Asia can appear to be an admix with Europe. And then finally, besides the exceptions described, undeclared root populations appeared at no greater than 15% of the prediction. So that would be, so for example, if someone said they were European, you wouldn't see African or East Asian popping up at greater than 15% in the data that we tested, in the samples that we tested. All right, so based on these results, as well as additional samples, including more admixed samples, we developed reporting guidelines, and these are our current guidelines that we have under consideration. So first off, for two of the root populations, we redefined the population names just to better reflect the region that they're referring to. So so that was, everything stays the same except for Africa. We now refer to it as Sub-Saharan Africa, and then Southwest Asia, in our reports, we'll refer to it as the Middle East. Okay, so I'm going to go through, as I say, the reporting guidelines. So there's 4 possible outcomes based on what we're considering right now. So the first one is a single inclusion. So that would be, in your report, you would be reporting one population. So this would be if the root population is greater than or equal to 80% and all the other root populations are less than 15. So if we look at an example, so this is a person that we tested and the results came back as 95% East Asian, 5% America, and we would report East Asian. So in this case, this particular person had South Korean ancestry. The next group would be is called a single mixed inclusion. So this, you're still reporting a single root population, but there is a bit of ambiguity in that there could be that this person could have a little bit of another ancestry, or they could be a little admixed. So this is when a root population is between 55 and 75%, and all the other root populations are less than 15%. So here's an example. So this person, when we tested them, the results were 75% South Asian, 10% African, 5% America, and 10% European. And so what we would report is that this person is South Asian or a mixed ancestry of a South Asian and at least one other root population. And this particular person, they reported that they're from Guyana, or their ancestors are, but that at least some of their grandparents are from India. So this does match up with what they did declare. There is a bit of ambiguity in their declaration, so we're not sure entirely what their ancestry is. But this does, as I say, does agree with the major part of their declaration. The next one is a double inclusion. So this would be when you're reporting two ancestries. So this would be, you would report 2 populations if they're greater than or equal to 20% and all the rest are less than or equal to 15%. So this is an example of the results where this person was 55% European and 45% Southwest Asian were the results. And we actually had, of the samples we tested, 5 of them had that prediction. But we would report that they were European or Middle East or a mixed ancestry of European and Middle East. And so here are the 5 samples that we that had those results. So the one person's Greek, the other was Italian, Romanian, the 4th person Polish, they were Ashkenazi Jewish ancestry. And then the fifth person is an admix of Greek ancestry and Turkish ancestry. Okay, and then the final category is we would report results are not determined. And this is when you get into more highly admixed samples. So you have 3 or more populations that are greater than or equal to 20%. And this is where, so this is an example of someone who their results came back as 30% European, 25% Southwest Asian, 20% East Asian, and 25% African. So we would report that the results are not determined. And this particular person is from South Africa, and they do have quite an admixed background. So his mother is South Asian, there's European, and the father is African and Europe as well. So, so you can see the person is highly admixed. So essentially, just to summarize and give you an overall idea, we would report an ancestry if it's greater than or equal to 20% for 1 or 2 populations. And then if you go more than 2 populations, then the results would become undetermined. All right, so the final group I said I'd come around to was the Americas. So this population is important for our population in Ontario. According to the 2011 census data, there are over 300,000 people in Ontario who identify or self-declare as being First Nations people. And it was important for us to test because, as you can see, these pins down here, the pink pins, these are the populations that were used to define the Americas. But you can see that Ontario is in North America, and these are in Central and South America. So we wanted to see what people in Ontario would look like, what their prediction would look like, and how accurate it would be. So unfortunately, we had a difficult time obtaining samples. So what we ended up doing is testing 10 samples in which the people declared that at least one of their grandparents had First Nations ancestry. So of those 10 people, 8 of them had results that included Americas at 25% or greater. So for those people, in our current reporting guidelines, we would report that they have, or part of the report would at least include that they were Americas with part of their ancestry, or if they had, in one case, we would report that the results were not determined because the person appeared highly admixed. The final 2 samples, their America's prediction came back as 10%. So 10% is no greater than the percentage of Americas that we saw in people that did not declare Americas in their ancestry. So in this particular case, we would not report Americas. So some possible reasons for why we're not seeing Americas pop up in their prediction. There's a few. So it could be that the populations that were used to define the region aren't the best populations to describe the populations we see in Ontario. It could also be that the donors don't have a complete or accurate knowledge of their ancestry. And then the final reason is kind of a combination of those two. First 2 points is that the 3 First Nation groups are identified as the first peoples to be in the land that we now call Canada. And one of those groups is the Métis group, and they are a mix of Americas and European. So there's already a history of admix beyond when Canada was formed in the First Nations people. So this could all come together to cause us to get low America's prediction. So that being said, so given all this, would our results be misleading if we said that these 2 people were not of? If we said that if we didn't include Americas in their results? So both donors also declared European ancestry, and Europe would be included in their report. And this brings us to the crux of this testing. So investigators are going to take the results we give them and deduce an appearance. So when we looked at the photos of the donors of these two samples, their appearance didn't disagree with the reported results. So we would say that they could appear to be European. So all that being said, one of the individuals in this group of two, they do live on a First Nation reserve, and this does lead us to some important points about how we communicate and release these results. And I mean, this topic could be a whole presentation on its own. But the idea of differentiating between genetic versus cultural ancestry. So the genetic ancestry that we report may be different with what someone identifies with culturally. So that leads into some of the other considerations when we're developing our guidelines and how we're implementing or releasing this data to our clients. So we acknowledge that this isn't like a regular, like our STR typing, that this service, we will require more training with our clients. So we are going to limit the service to client groups that have had orientations to the testing. We're also going to invite investigators to meet with us to discuss the reports and answer any questions. And at that point too, we will have, as I mentioned, we will have some training around what the results mean. And then we're also ensuring that the reports are developed to be as clear as possible. Okay, so I'm going to switch gears just a little bit and talk about, just touch on some of the forensic type samples that we tested, because this whole validation was not just about testing the model, the ancestry prediction model. It was also about, you know, a new sequencing technology and how do forensic type samples work within that system. So I'm going to talk about some mock forensic samples. I'm going to talk about mixtures and then some degraded samples. So for the mock casework samples, what we did is we obtained blood from a variety of sources from our volunteers, this type of DNA you would see in casework, and then we applied it to a variety of substrates and then extracted it in 3 different extraction methods. So phenylchloroform and 2B-based extraction methods, the easy one, and DNA IQ. Prior to running it on the MPS, we ran it through our current casework workflow. So we were able to look at the qPCR plots as well as the CE profiles and determine that there was no degradation in these samples and there were no detectable inhibitors based on our current workflow. And what we saw is that the genotype results and all the quality metrics associated them were comparable to samples that we had run from the same donors in which we were just using good quality buccal swabs to run, because that's all we had to compare to. So basically we had no issues running these samples. So the genotype was completely concordant, quality metrics were good, and obviously then the ancestry predictions were the same as in the other runs. So we also ran some mixtures, and one thing to note is that this software currently doesn't allow you to deconvolute mixtures and put that deconvoluted profile back in the software to get an ancestry prediction. So what we were left with was trying to find the point in the mixture ratio at which you're just detecting the major contributor. So we did a few, but I'm going to just show you one in which the major was 100% African and the minor was 100% East Asian. So this graph shows you the results. So on the x-axis is the mixture ratios, and then on the y-axis is the percent African. So we're wanting to see the point at which you're seeing 100% African. So you can see here that at 16:1 and 32 to 1, we're seeing the major contributors' ancestry. But I want to also point out, though, that with that, so the numbers along the top are the number of genotype discordance between the major predictor, majors genotype and the mixtures genotype. So you can see that we did, even though we had discordance at 2 loci in one and 1 at the other, that we still did get the major contributors' ancestry predicted. And then finally, we ran some degraded samples, and this is just a subset of the samples we ran. But I just want to point out, I think Catherine already pointed out that the ancestry panel has the amplicon sizes between 76 and 199 base pairs. So it is, the amplicons are smaller in size than what we have with our ID+. So it's, hopefully you'd expect it to perform better with degraded samples. So our samples, we had in the first, can't move the arrow here. In the first 2 columns, so this is, or in the second column, this is the number of loci with dropout in the ID+ kit. So you can see we tested ones with 2 all the way up to dropout at 10 loci. And then these top 2 in brackets, it just shows the number of loci with dropout where the loci is less than 200 base pairs. And then the next 2 columns are the MPS results, and this one has the discordant genotype calls and the number of no calls, or the number of calls that we had when you compare it to the good quality comparison that we had. And what you can see is that most of them we are getting, we don't have any discordant calls, and we are getting most of the SNPs, or all of the SNPs being called, with the exception of these 3 samples. So these 3 samples, there was between 2 and 4 discordant genotype calls, and then these ones had dropout at between 10 and 2 loci. So despite that, all of them had, the ancestry results were correct for what was, when compared to the declared ancestry. Okay. So that's just giving you a flavor or a little taste of the validation, what we did. We are planning to implement this service early this year. And I just want to acknowledge the people that worked on this. So Solby Jin was involved from the beginning on the validation. Then we have James Morrow, Margaret Henry, Sobia Malik, and Gary Alderson, which have been developing the more client-facing components of this. So they've been working on guidelines and client training materials. And then the steering committee, Joel McGrory, Jonathan Millman, Neil Fernandopoul, and Jack Laird, who have been directing this project. And at the bottom, you can find my contact information.
All right. Thank you so much, Moretta. We are going to move into our last presentation of the day. This one's going to be a little bit different than really anything that we've ever done here with our webinars. So due to time zone restraints, Dr. Runa Daniel is actually in Australia, so she is about 16 hours ahead of East Coast. So we were able to record her presentation, which we are going to play for you now. Please make sure that you are on a landline if possible. That will give you the best performance for the video. And without further ado, I'm going to go ahead and start today's final presentation for the SNP webinar. Again, if you do have any technical issues, just let us know in the chat pod and we will work with you to get those resolved.
Hi everyone, I'm Runa Daniel and I'm going to talk to you today about DNA intelligence, application and considerations. So before we go on, I'd just like to let you know that this presentation cannot be retained, reproduced or recorded without consent, and that screen capture of the slides is not permitted. So we'll begin by looking at the process of DNA profiling, which you're all familiar with. And this is where we retrieve biological evidence from crime scenes, extract the DNA, generate an STR profile, and search the database using this profile. In the cases where a database match is obtained, this will lead to the identification of the donor of the biological material. However, in the cases where a DNA profile match is not obtained, for the majority of operational labs, this is where the use of biological evidence ceases. This may also be the case if a non-informative partial profile is generated, or if an STI profile is not generated, possibly due to severely degraded or compromised samples. In these circumstances, the investigators are left asking who is the donor of the biological evidence and what do they look like. Forensic DNA intelligence is the inference of biogeographical ancestry and externally visible characteristics from biological evidence. The inference of biogeographical ancestry, or BGA, involves the differentiation of individuals from major global populations and also from subpopulations within this region. And forensically relevant externally visible characteristics include features such as eye, hair and skin colour, baldness, hair morphology and age. So what we're talking about here is looking at biological evidence beyond just for identification, which is how the majority of labs use this evidence currently, and looking at extracting valuable intelligence information associated with the donor of the biological evidence. So what we're talking about really is a paradigm shift away from just analysing this evidence for identification purposes and to be able to generate valuable intelligence and then feed this intelligence back into the investigation to assist with an identification. So forensic DNA intelligence can be used to create or narrow a pool of suspects. It can be used to provide investigative leads in the primary and critical stages of an investigation. It can also be used for mass disaster victim identification, for example, plane crashes where a manifest of individuals is available, but also in mass DVI where a manifest of individuals is not available, for example, floods, hurricanes, fires, and tsunamis. And forensic DNA intelligence can be used to replace notoriously unreliable eyewitness statements. And this is an example with the Louisiana serial killer case in 2004. And this is where 7 women were killed between 1992 and 2003. In one of the cases where a young female college student was murdered, an eyewitness reported seeing a white Caucasian male in the vicinity of the crime scene. And this is a composite sketch that was released based on the eyewitness account. So the investigators focused all of their resources on collecting DNA samples from white Caucasian males in the vicinity, but none of the STR profiles generated from these males matched the evidential profile. A year passed and there wasn't any progress on this case, so the investigators submitted the sample to DNA Print Genomics, which was the first commercial provider of ancestry and phenotype testing. And the results indicated that the donor of that evidential material was 85% sub-Saharan African and 15% Native American. That their skin shade was to be average to darker than average in tone, and they stated that the probability of this donor being European was low. So automatically, what you can see is from the description of the data, this is the exact opposite to the description provided by the eyewitness account. Based on this data, the investigators' memories were triggered of an individual in the area that had been arrested for peeping and burglary, and they paid this individual a visit and obtained a DNA sample, and sure enough, this individual's STR profile matched the evidential profile, and Derek Todd Lee was arrested and charged with murder. So if you have a look at the composite sketch based on the eyewitness account, this description would never have led the investigators to the donor of that biological evidence. So if you are looking to implement DNA intelligence into your case workflow, how would that work? Well, at the point where the STR profile is not informative, the sample would then go into the DNA intelligence workflow, and this is then going to possibly, from the data generated, lead the investigators to a suspect. Once this happens, they need to be able to obtain a DNA sample from the suspect, from which an STR profile needs to be generated and then compared back to the evidential profile. So what we did here at the Victoria Police Forensic Services Department was we engaged in a DNA intelligence exercise, and this is where we provided DNA intelligence to police investigators and intel analysts. So why did we do this? Well, we know how useful DNA intelligence can be in an investigation, but what do the investigators actually think about this sort of intelligence, and would it be useful to them? If so, what do the investigators need from us forensic biologists? And we wanted to know whether a police intel analyst was able to interpret or understand an ancestry analysis report that was provided by the forensic biologist. So in this exercise, we've focused mostly on ancestry informative data. So by working with police intel, it provides us with an opportunity to improve the development and reporting of intelligence tools. So this is the DNA intelligence exercise, and this is what we provided to one of our police intelligence analysts. We created a mock case and we provided excerpts from the case report, as well as the victim statement, the forensic report, and then the ancestry analysis report. And what we did here was that we provided the ancestry analysis data to forensic biologists who have expertise in interpreting the data. And the reason we did this is because we also wanted to have a look at the variation in terms of how these biologists interpret and report the ancestry data. We also wanted to start to understand what the investigator or the intel analyst will understand from the biologist's reports and what type of reporting and expression was useful to the intel analyst who is not a biologist. We also provided a suspect pool of photos, and further on in the exercise, we provided some additional DNA intel, which was about externally visible characteristics, including age. And the task of the intel analyst was to apply the DNA intelligence to the... This is our Victoria Police Intel Analyst, Sergeant Anthony Lawler. He's a Senior Intelligence Analyst and Intelligence Coordinator in our Crime Intelligence Group here at Forensic Services at Victoria Police. So Sergeant Lawler has worked in investigation units, including the Specialist Crime Task Force, and in a number of police intelligence roles, including managing frontline intelligence teams. And Sergeant Lawler has been in crime intelligence here with us since 1993. So this is the mock case of the bike bandit, and this is an excerpt from the police report of the victim's statement. The victim stated that he was attending the 23rd International Congress of the We Love DNA Society forensic meeting at the Grapes R Us Winery in the Yarra Valley, Victoria on the 1st of April 2016. Approximately 15 people attended the meeting from Victoria Police Forensic Services Department and the National Institute of Forensic Science, and all attendees were accommodated on site. The bike was placed in a bike rack unsecured outside the meeting room. The victim reports that the bike was last seen at 1800 on the 1st of April when he was departing for a social event that evening on the premises. The victim states that when he went to collect his bike at 0800 on the 2nd of April, he found that the bike had been taken. The victim contacted the police on the 2nd of April at 0830 to report the theft of his bike. Forensics attended the scene at 1100 on the 2nd of April. A red colour stain was found on the bike rack where the bike had been leant against. The stain was collected and submitted to the Biological Evidence Branch at Forensic Services Department. No additional forensic evidence was retrieved. 6 suspects were yet to be excluded. So this is a photo of the stolen bike on the Yarra River here in Melbourne. And this is a photo of the victim on his bike in his hometown of Santiago de Compostela in Spain. So our good friend Chris Phillips has had his bike stolen. Excuse me. This is an excerpt from the forensic analysis report, and this is indicating that a single source female profile was generated from the red stain and that no DNA database matches were obtained, so the DNA extract was submitted for ancestry analysis. The ancestry analysis was done using the NGS kit Precision ID Ancestry Panel by Thermo Fisher Scientific, which you would have heard about today from Catherine. So as you know, it generates a heat map, which you can see on the left-hand side, and it indicates the admixture proportions from the 7 root populations, which are used by Genotyper to provide this ancestry profile. And what you can see from the admixture proportions is that the individual is 85% European and has a 10% South Asian and 5% East Asian component. So what this is indicating to us is that this is an admixed individual. On the right-hand side, what you can see are subpopulation likelihood estimates. Now, according to the manufacturer, if the admixture proportions indicate that it's an admixed individual, then the subpopulation likelihood estimates are not accurate. So for the purposes of this exercise, we just based the ancestry intelligence on the the admixture proportions, indicating that the person has the majority European ancestry with the South and or East Asian minor component. So remember that we wanted to provide this ancestry data to forensic biologists who have expertise in interpreting this data. And we wanted to compare the variation in terms of interpretation between these biologists and also the variation in how they would report this information. So we'll go through each of the reports. Expert 1 stated that there is a high likelihood that this individual has a majority European genetic ancestry. However, there is also a high likelihood of a minor contribution from South and or East Asian genetic ancestry. As a result, there are 2 possible interpretations in no particular order. The individual has a majority of ancestors from Europe and a minority from South and or East Asia. For example, 3 grandparents from Europe, 1 grandparent from South and or East Asia. Second, the individual has ancestors from a region or regions intermediate between Europe and South and Eastern Asia, but genetically closer to Europe, for example, from Eastern Europe or the Middle East. Expert 2 stated something very similar, but a revised version, also indicating that the individual has a majority of ancestors from Europe and a minority from South and or East Asia, or that the individual has ancestors from a region or regions intermediate between Europe and South and Eastern Asia, but genetically closer to Europe, for example, Eastern Europe or the Middle East. And this expert also stated that it's not possible to distinguish between these 2 possibilities. And as you can see, expert 3 was completely different, stating that the DNA sample donor is 320 times more likely European than Middle East in origin. So already what we can see here is that there's quite a bit of variation in the way that the biologists are going to interpret and report this ancestry data. So the next step was to get Sergeant Lawler's feedback as the Intel analyst on the biologist's report. So as he was going through the reports and providing feedback, we were recording our observations. Of expert 1, he stated that it was too much information, that it created confusion. He said he felt bamboozled. So bamboozled is an Australian slang term which indicates that he felt overwhelmed by the information. He stated, we need more concise information. Who done it? Who are we looking for? Perhaps provide the extra information only if requested. Of expert 2, he stated that it was a good amount of information, and of expert 3, he said it was very concise, we may need more information. So he concluded that a combination of the report from expert 2 and expert 3 would be good. So Sergeant Lawler is now about to apply this information to the suspect pool. And before we go on, I'd just like you to note that the participants in this mock case have not and are not involved in any criminal activity. So this is the suspect pool, and Sergeant Lawler's task now is to apply all of the information that he's been provided in this mock case, as well as the ancestry reports from the biologists, to this suspect pool. And we were recording our observations of how he was applying this. So one by one, he started to consider the suspects that may fit this ancestry profile. He thought it could possibly be a suspect number 1, suspect number 5, possibly even suspect number 4. And so for the next little while, he started to group these photos based on who he thought were more likely to fit that profile and who he thought were less likely to fit that ancestry profile. So he kept grouping these, and this went on for probably 20 to 30 minutes. What was also interesting was during this period of time, he actually kept referring back to the output from the Precision ID Ancestry Panel genotyper analysis. And when we asked him why, he said that having this geographic map, as well as the root populations and the proportions, he felt assisted him in understanding the biologist's report and the ancestry profile of this donor. So this was a surprise to us because we just assumed that he would base his application of this information purely on the biologist's report. But in fact, he seemed to like this as a visual aid. And as I said, having the map and the numbers seemed to assist him in understanding the ancestry of the donor. So at the 30-minute mark, we said to Sergeant Lawler that we also have some additional DNA intelligence, and that's about the externally visible characteristics of the donor of this evidence. And we stated that the eye colour was hazel brown and that the hair colour of this individual was brown. And with this information, at the 30-minute mark, he immediately excluded suspect number 2. So after another 5 minutes, we then stated to Sergeant Lawler that we've also generated some information about the age of the individual and that the age range of this donor was 42 to 48. So immediately Sergeant Lawler excluded another 3 suspects. So here we are at the 45-minute mark, and Sergeant Lawler, just by sitting at a table with 6 photos and the information about the case, was able to reduce this suspect pool of 6 down to 2 individuals. So we had a discussion about what the next steps would be, asking what he would do, and he indicated that he would progress the investigation focusing on these specific suspects. And we explained that they would then need to obtain a DNA sample from the suspect and compare this suspect's profile back to the evidential STR profile. So Sergeant Lawler then stated that at this point they would consider collecting a covert sample if that was in a real case. So in this mock case, that's what happened. And he generated, so from this suspect pool that's been reduced down to 2, the STR profile that was generated from one of these 2 suspects actually did match the evidential profile. So it matched the evidential profile of suspect number 4. So well done to any of you who, while listening to this, did select number 4. And this also indicates how powerful this tool is because Sergeant Lawler has correctly narrowed down a suspect pool of 6 down to 2, and within the 2 was the donor of the evidential material. So the next thing was we wanted to look at the ancestry profile and see how accurate it was for this donor. So this donor is actually Dr. Lindsay Wilson-Wild, who's the director of our National Institute of Forensic Science. So remember that the ancestry data was indicating that she has a majority European ancestry, but she also has South Asian, possibly a minor East Asian component. Lindsay's maternal grandfather is English and her maternal grandmother is Ukrainian. So immediately we see where the European component is coming from. Lindsay's paternal grandfather is Indian with Dutch admixture, and her paternal grandmother is Indian. So we're seeing where the South Asian component is, but remember that the paternal grandfather is Indian with the European component. So the ancestry profile generated using the Precision ID ancestry panel was correct and accurate. So we repeated this DNA intelligence exercise, this time with 9 senior intelligence analysts and investigators from the Victoria Police Major Crime Squads. So I won't go through all the details, but what I will do is provide you with a summary of the outcomes of the 2 exercises combined and what our observations were. So of the 2 exercises combined, the suspect pool was reduced from 6 to 2 individuals in 45 to 60 minutes. So this is a significant result because in a real case, the investigators would have had to pour a number of police and forensic resources into being able to exclude suspects from their suspect pool, and that could take days to weeks depending on what their resources were. But in contrast, by sitting at a desk with this DNA intelligence and a photo pool of suspects, they were able to reduce this suspect pool down to 2 in